Welcome back, everyone. Ready to dive into something pretty fundamental to life as we know it.
Oh, absolutely. You know me, always up for a deep dive into a good metabolic pathway. And today it's all about the citric acid cycle.
The citric acid cycle. Talk about the heart of cellular metabolism.
No kidding. We're talking energy production, we're talking biosynthesis. It's the hub where carbs, fats, and proteins all come together to fuel the cell.
Okay, so for our listeners who maybe haven't thought about the citric acid cycle since their intro biochem class, let's set the stage here.
Great idea. You want the quick and dirty.
Exactly.
All right, so it starts with acetyl-CoA.
Acetyl-CoA, which comes from breaking down sugars, fats, you name it.
Right. And the cycle takes that acetyl-CoA and oxidizes it. Strips it down, essentially, to generate these high-energy molecules, NADH and FADH2.
And those are the real stars of the show, wouldn't you say?
Absolutely. They're the ones that head off to the electron transport chain, the cell's powerhouse. That's where the bulk of ATP, the cell's energy currency, gets made.
So the citric acid cycle is all about extracting energy from fuel molecules and setting the stage for oxidative phosphorylation.
It's a multi-step process, very carefully orchestrated. But here's the kicker. It doesn't just make energy.
Oh, yeah, that's right. It's also a source of building blocks for other important molecules in the cell.
Totally. We're talking amino acids, for hemoglobin, even fatty acids. It's a true metabolic chameleon.
Amphibolic, as they say in the textbooks.
Yeah.
All right, enough of the overview. Let's get down to the nitty-gritty.
You got it. Time for a step-by-step breakdown of this metabolic masterpiece.
Perfect. So step one, if I recall correctly, starts with our two-carbon friend, acetyl-CoA, meeting up with oxaloacetate.
4 carbons in oxaloacetate, so we're already at 6 carbons total. And their meeting point, citrate synthase.
Citrate synthase. Classic enzyme. What's special about it, though?
It's all about the induced fit mechanism. This enzyme, it's not just some rigid lock and key.
So it doesn't just sit there passively waiting for its substrates.
Not at all. Citrate synthase is dynamic. It changes shape as it binds acetyl-CoA and oxaloacetate, bringing those reactants in close, facilitating the reaction.
It's almost like a handshake that gets firmer as the molecules get closer.
That's a good analogy, a dynamic embrace. And boom, you've got citrate, the first product of the cycle.
6 carbons in citrate, just like we were expecting. Okay, step 2. This is where it gets a little tricky, right?
You're talking about the conversion of citrate to isocitrate.
Yep, and that whole procurality thing always threw me for a loop.
I get it. Citrate, on the surface, it looks symmetrical, but it's got this hidden asymmetry.
It's like a pair of gloves. They look the same, but try putting the right glove on your left hand. Doesn't work, does it?
Perfect analogy. And the enzyme aconitase is the one that sees this difference. It only acts on one specific form of citrate, leading to isocitrate.
So a lot of specificity there. What happens if something messes with this step?
Oh, well, there's this nasty toxin, fluoroacetate. You've probably heard of it.
Rings a bell. So to do with pesticides.
That's the one. It gets metabolized into fluorocitrate, which then binds to aconitase, and that, well, let's just say it doesn't end well for the cell.
Shuts down the whole cycle.
You got it. A single fluorine atom just thrown into the mix, and the whole thing grinds to a halt. Crazy, right?
Shows you how fragile these pathways can be.
Yeah.
Okay, on to step 3. What have we got?
Step 3, we're talking isocitrate to alpha-ketoglutarate, our first oxidative decarboxylation.
Oxidative decarboxylation. So we're losing a carbon and something is getting oxidized.
You bet. Isocitrate dehydrogenase is the enzyme here, and it's a key regulatory point. Generates NADH, our first energy carrier of the cycle.
Right. So we're starting to see the link between the citric acid cycle and the electron transport chain.
Exactly. It's all connected. And just to add a little wrinkle, some isocitrate dehydrogenase enzymes use NADP plus instead of NAD plus S.
So the cycle can be tuned to produce different electron carriers depending on the cell's needs.
Exactly. NADPH is more involved in biosynthesis, while NADH is primarily for ATP production. The cycle is a multitasker.
Love that. Okay, step 4. Another oxidative decarboxylation, if I'm not mistaken.
You are not mistaken. This time, it's alpha-ketoglutarate getting transformed into succinyl-CoA. And guess what?
Another big enzyme complex.
Ding, ding, ding. We're talking alpha-ketoglutarate dehydrogenase complex. Another multi-enzyme behemoth, just like pyruvate dehydrogenase.
So nature likes to reuse its best tricks, huh?
You bet. And just like with pyruvate dehydrogenase, we get another molecule of NADH. Plus, we end up with succinyl-CoA, a high-energy thioester compound.
Thioester bonds. Those always mean business when it comes to energy.
Oh, yeah, they pack a punch. And keep in mind, we've lost 2 carbons as CO2 at this point.
Right. Started with 6 in citrate, down to 4 now in succinyl-CoA. Making progress.
We are. And step 5 takes us to a really exciting moment. This is where we get substrate-level phosphorylation.
Meaning?
Meaning we're making ATP directly, or GTP, depending on the tissue.
So up until now, it's been all about those electron carriers. But now we're finally cashing in some of that energy.
Exactly. And the enzyme responsible for this magic, succinyl-CoA synthetase.
Catchy name.
It breaks that thioester bond in succinyl-CoA, releases energy, and uses it to make ATP or GTP. It's like a little power plant within the cycle itself.
And different tissues make different nucleotides. That's interesting.
It's all about tailoring energy production to the specific needs of the cell. The heart, for example, mostly makes ATP, while the liver leans towards GTP.
Fascinating stuff. So we've got ATP, and our 4-carbon molecule is now succinate. But we still need to get back to oxaloacetate to complete the cycle, right?
Absolutely. We've got 3 more steps to go, each with its own twists and turns. But maybe we should give our listeners a moment to catch their breath before we keep going.
Good idea. There's a lot to digest here. Okay, folks, back to the citric acid cycle we go. Where were we? Ah, yes, succinate. 4 carbons, ready for action.
And ready for some serious transformations to get us back to that oxaloacetate.
Right, because we need to keep that cycle spinning.
Mm-hmm.
So what's next on the agenda?
Well, get ready for some redox chemistry, a little hydration, and even a touch more energy extraction.
Sounds like a party in a mitochondrion.
It kind of is. Okay, so step 6. This is where succinate dehydrogenase steps into the spotlight.
Succinate dehydrogenase. I remember that one. It's the one embedded in the inner mitochondrial membrane, right?
That's the one. It's a bit of a celebrity, actually. Has a dual role in the citric acid cycle and the electron transport chain.
Talk about multitasking.
Oh, yeah. It's a busy enzyme. So it takes succinate, oxidizes it to fumarate, and in the process, it reduces FAD to FADH2.
Okay, so FAD is our electron carrier here. Why not NAD plus J? Like in the other steps?
Good question. It has to do with the energy involved. Turning a single bond into a double bond, like we do here, requires a stronger oxidizing agent. FAD is up to the task.
Ah, so FAD steps in when we need a bit more oomph.
Exactly. And the FADH2 produced, it doesn't just float around. It's directly fed into the electron transport chain, thanks to succinate dehydrogenase being part of complex 2.
So efficient. It's like a metabolic assembly line.
Nature at its finest. Now, here's another cool thing about succinate dehydrogenase. It's stereospecific.
Stereospecific. So it's picky about which form of succascent it acts on.
Very picky. It only removes hydrogens from specific positions, leading to the trans isomer fumarate. No cis isomer allowed.
So the enzyme ensures we get the right product. No unwanted side reactions.
Exactly. Keeps everything running smoothly. Okay, step 7. Time to bring some water back into the mix.
Hydration time. So fumarate gets hydrated to form malate.
Right, you got it. And the enzyme in charge of this transformation is fumarase, also known as fumarate hydratase, if you want to get fancy.
Fumarase, got it. And is this step stereospecific too?
You bet. Fumarase adds water across the double bond in a very specific way, resulting in only the L isomer of malate.
Wow, so much control over the stereochemistry.
Yeah.
These enzymes are amazing.
They really are. It's all about precision and efficiency in these metabolic pathways.
Okay, we're in the home stretch now. Step 8, the grand finale, takes us back to our starting point, oxaloacetate.
And malate dehydrogenase is our star player for this last act.
Malate dehydrogenase. It oxidizes malate back to oxaloacetate, right?
Precisely. And it generates our final molecule of NADH in the process.
Perfect. We've regenerated our 4-carbon acceptor molecule, topped up our NADH stores, and completed one full turn of the citric acid cycle.
A masterpiece of metabolic choreography, wouldn't you say?
Absolutely. But I have to ask, how does the cell keep this whole intricate dance in check? I mean, the energy needs of a cell are constantly changing.
Ah, excellent question. And that's where things get even more fascinating. Because the citric acid cycle isn't just a static set of reactions. It's constantly being modulated, fine-tuned by a whole network of regulatory mechanisms.
Regulatory mechanisms. So we're talking feedback loops, allosteric regulation, that kind of thing.
Oh, you better believe it. And we're just getting started. But maybe we should pause here again and let our listeners soak it all in.
All right. Time to unravel the mystery of how the citric acid cycle stays in sync with the cell's ever-changing energy needs.
It's all about regulation, right? keeping things running smoothly, responding to signals.
Exactly. So where do we even begin with something as complex as this?
Well, we could start by looking at the entry point. How does the cell control the flow of fuel into the cycle in the first place?
Right, because it all starts with acetyl-CoA.
And the gatekeeper there is pyruvate dehydrogenase. Remember that giant complex we talked about? The one linking glycolysis to the citric acid cycle?
Oh yeah, can't forget that one. Talk about a metabolic crossroads.
No kidding. So pyruvate dehydrogenase, it's incredibly sensitive to the levels of its products, acetyl-CoA and NADH.
Ah, so when those products start building up, that's the signal that the cycle has enough fuel.
Precisely. They act as feedback inhibitors. Basically, they tell pyruvate dehydrogenase to chill out, slow down, prevents a metabolic traffic jam.
Makes sense. Conserves resources. But didn't you mention something about covalent modification too?
Right. There's another layer of control. It involves phosphorylation.
Phosphorylation.
Yeah.
Like adding a phosphate group to the enzyme.
You got it. When energy levels are high, a specific kinase comes along and phosphorylates pyruvate dehydrogenase, switches it off, essentially.
So it's like putting a pause on the cycle when ATP is plentiful.
Exactly. Prevents the cell from wasting glucose when it doesn't need to.
But when energy levels drop, we need to get that cycle going again, right?
Enter the phosphatase. It removes that phosphate group, reactivating pyruvate dehydrogenase. Back in business.
It's like a constant tug of war between the kinase and the phosphatase, fine-tuning the activity based on the cell's energy status.
Like a metabolic thermostat. And it's not just ATP levels they're responding to. Calcium, insulin, all sorts of signals can influence the balance.
Wow, so much complexity.
Yeah.
So we've got the entry point covered. What about regulation within the cycle itself?
Oh, there are key checkpoints within the cycle, too. Remember those 3 reactions with the big negative free energy changes?
The ones that really push the cycle forward. Citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase.
Yeah. Those are the ones. Prime targets for regulation. And a big factor here is the NAD plus NADH ratio.
The redox state of the cell is like a measure of how much energy is available.
Exactly. High NAD plus means the cell needs energy, so the cycle speeds up. Low NAD plus, plenty of energy, the cycle slows down.
Like a gas pedal and a brake pedal for metabolism.
I like that. Now, let's focus on isocitrate dehydrogenase for a minute. This enzyme is a bit of a control freak.
In what way?
Well, it's allosterically activated by ADP.
ADP. So when ATP levels are low, ADP levels rise, and the cycle gets a boost.
You got it. But here's the twist. It's also inhibited by ATP and NADH, its own products.
Talk about a feedback loop. Keeps everything in check.
And alpha-ketoglutarate dehydrogenase, it's got a similar setup, inhibited by its products succinyl-CoA and NADH.
So it's all about balance.
Yeah.
Feedback inhibition, the NAD plus NADH ratio,
Yeah.
and even just the availability of substrates. It's all connected.
A beautifully orchestrated system. Now, there's one more molecule we need to talk about, citrate. It has a regulatory role beyond just being an intermediate in the cycle.
Oh yeah, I remember you mentioning that. Something about its connection to glycolysis.
Right. Citrate can actually leave the mitochondria and head out to the cytoplasm.
A little field trip. What does it do out there?
Well, in some tissues, like the liver, it can inhibit phosphofructokinase. That's a key enzyme in glycolysis.
So when citrate levels are high, glycolysis slows down, keeps things in sync.
Exactly. It's a way to coordinate these 2 crucial pathways, making sure they're working together efficiently.
Amazing how everything is so interconnected. Now, what about citrate synthase? Didn't it used to be considered a major control point?
It did, but recent research suggests that it operates pretty close to equilibrium. So substrate availability, especially the concentration of oxaloacetate, seems to be more important in regulating its activity.
So it's not just about enzyme kinetics. It's also about supply and demand.
Precisely. And that brings us to a rather sobering thought. What happens when this finely tuned machinery breaks down?
Right, because we've talked about how important the citric acid cycle is. What are the consequences when things go wrong?
Well, unfortunately, defects in these enzymes have been linked to a whole range of human diseases.
Like what?
Well, mutations in succinate dehydrogenase, fumarase, and alpha-ketoglutarate dehydrogenase, for example, have been implicated in neurodegenerative diseases.
So these metabolic errors can have serious consequences for brain function.
Absolutely. And it's not just neurological disorders. These enzyme defects have also been found in tumors, suggesting a link between altered metabolism and cancer.
Wow, that's really concerning.
Yeah.
So a malfunctioning citric acid cycle can have far-reaching effects on human health.
It really highlights the importance of this pathway, not just for understanding basic biology, but also for developing new treatments for these devastating diseases.
Well, I think we've covered a lot of ground today, from the intricate steps of the cycle to the elegant regulatory mechanisms and even the consequences when things go awry. It's been a fascinating journey through one of the most fundamental processes in life.
Absolutely. And it's a reminder that even at the cellular level, there's so much complexity and beauty to uncover. It's what makes biochemistry so captivating.
To our listeners, we hope this deep dive has sparked your curiosity and inspired you to explore the world of cellular metabolism even further. Until next time, keep those brains buzzing.